So I left the title pretty general because I hadn't fully decided what I wanted to talk about. But in the end, I decided to go for mostly description of two new pulse sequences that we've developed applied to structure determination for magic angle spinning NMR. And I'll start with the acknowledgement slide. This is a slightly outdated picture of the group. Mostly the work will be that I'll present today is from Evgeny Nemirovsky and also Kai Sue, who are two postdocs in the lab. So a brief outline, I'll first give a little bit of motivation for our work since we're interested in membrane proteins. And then most of the talk will be about a selective recoupling sequence. And finally, a little bit about our new diagonal suppression technique. So these are the membrane proteins that we're interested in. And in the interest of time, I don't want to say too much about these, but I'll just put them up on the slide in case you have an interest in them and discussing later about these particular membrane proteins. And I just wanted to say, by way of motivation, that even in the days of AlphaFold, we've still found a lot of interesting topics in these membrane proteins, as I'm sure many of you all have as well. And these are three examples showing measurement of detailed chemical bonding, hydrogen bonding, and active site of membrane protein at functional residues of this proton channel. Detecting water, in this case, a really stably bound water that exists for milliseconds where we could measure its chemical shift at 10 ppm. And of course, also measurement of dynamic parameters. And for this work, what's maybe more relevant is the topic of aggregated proteins or non-native protein states, which are more difficult to access. This slide just shows the progress over the years in this area of structure determination. In blue here are helical proteins, and in red some beta barrels. And you see the initial pioneering work of oriented samples represented in the many helical proteins that have been solved, and more recently moving towards larger and larger proteins, such that these days we can reach sizes into close to around 30 kilodalton and determine global structures. And I just wanted to highlight three of protein structures that I've been involved in solving recently. These are all beta barrel systems. And for these, it was really critical to have proton detected NMR and fast spinning at around 60 kilohertz or so and 1.3 millimeter rotors. And that may be a little bit surprising in light of some of our earlier work where we were interested in getting rid of the requirement for deuteration, which is quite important for membrane proteins, which can be difficult to deuterate. So we had shown the structure determination of this 14 kilodalton protein. It's a capsid protein. So this is not a membrane protein, but a capsid protein. Here we relied on many side chain contacts for the structure determination. And I just wanted to point out here that we were actually missing a lot of the contacts that you might think define the structure in these anti-parallel beta strands. So here highlighted in red are all the missing contacts that we had in these anti-parallel beta strands. We did measure a few of them in black, but more so we relied on side chain contacts for this structure determination. And of course, we'd also like to go to much larger proteins into the 30 kilodalton kind of range. And so the reason that we were missing these contacts is fairly simple, but I think I just wanted to highlight this problem because I think we often throw around the term a dense network of proton couplings, but just how dense is it? Well, if we look just at 5 angstroms, we already have about 25 protons rather in this is a fibril structure, but it's pretty consistent across different PDBs. We have about 25 protons, 5 of them are amides. And so if we have a broadband recoupling technique, we'll just spread the polarization out among all of those 25 protons and have hopelessly low sensitivity for longer distances. And if we go out to 8 or 10 angstroms, now we're talking hundreds of protons in that volume. And so this motivates selective recoupling techniques for the fully protonated proteins or deuteration of the side chains. So this shows one of the structures, a close-up from the VDAC structure, which was the work of Esther. And you can see that the structurally interesting contacts are above 3 angstroms. Meanwhile, in the side chain, there are many proton-proton distances which are below 3 angstroms. And all the way down to basically the van der Waals contact around 2.2 or so angstroms. And so predictably then, if we have a broad-banded recoupling sequence, the polarization just goes right out to the side chain. And this you can see here in red for RFDR, which is broad-banded, the polarization quickly is lost out to the side chain. So this motivated our development of a selective recoupling technique. We wanted a zero quantum technique so that different transfer pathways through relayed transfers would add up. This shows the design of the sequence with jump return elements of 45 degrees with the following phase cycle, which was found empirically in the computer. And I like to say that we found this pulse program by working at a very high level of abstraction, which is a fancy way of saying that we really didn't know how to design such a pulse program. So we just let the computer do the work. And so this is how we set it up. We had 4 protons, 2 of which are amide protons whose frequency was stepped across the amide band with a slightly longer distance than a nearby proton, which basically models an alpha proton. And then we varied the number of pulses per rotor period and their phase and looked for sequences that retained the total signal, which in a way is a filter against double quantum sequences, and provided efficient amide-amide transfer while essentially resulting in no transfer to the alpha proton. And this is how we ended up with a zero quantum first order recoupling sequence. So this sequence can be compared to some other sequences in the literature, such as SPR or SERP or BAS-CP. And we really were inspired by the work of Junyoung and his group with SPR in basically the search space for this pulse program. But we have a zero quantum sequence due to the phase cycle, whereas what they have done is double quantum sequences. So the double quantum sequences are really useful for quantifying the distances for the shortest proton, but the zero quantum is working better for overall transfer. And in the case of BAS-CP, it's a second order sequence, so it depends on a third proton, and we were really looking for something that would be first order, depending more on the distance and not a complicated 3-spin geometry. So we've implemented this into a 3D sequence, NHH-NH type of transfer pathway. This was recorded at the 1.2 gigahertz for the model protein SH3, just to see how this was working. And we can record quite a lot of contacts, up to 8 here, which indicates that we're reaching the kind of distances that we want to see. So these strips are looking fairly close to, for example, what Rasmus had published for deuterated proteins using RFDR several years ago. So this seems to be working pretty well. We can also compare with radio frequency driven recoupling for a deuterated protein, just to see which peaks we're retaining, how strong they are, and which we're missing. And this also compares very favorably. We basically observe the same cross peaks when the frequency separation is below around 2 ppm. And you can see this red strip here where we miss contacts because the frequency separation is just too large. But if we would search for a sequence that had a larger band, this would start to leak to H alpha too much. So this is, we think, a pretty optimal sequence. There are also, although you can't see in this overlay, there are also a few peaks which show up better in this sequence just because probably we've removed the losses to all of the other sites. So this is working pretty well for protein structure determination. I wanted to look at a few more characteristics of the sequence and also see if we can understand how it's working. So first off, it's not a band selective sequence. By that I mean that it has a minimal carrier frequency dependence. So it's really the frequency separation between the two recoupled spins and not a frequency band, which is excited. So this means the sequence could be extended into kind of multiple recoupling of, say, aliphatic plus aliphatic, and also amide to amide at the same time, but we haven't done that yet. And then we can look at operators to try to see the transfer pathway, which occurs in this. So this is starting with the single spin operators, detecting single spin operators in a simulation of just 2 spins. And as expected, if we set the offset to 0 for both spins in blue, we see the transfer. If we set the offset to be a significant value between the 2 spins, the offset difference between the 2 spins, then we don't observe the transfer anymore. But the transfer is occurring probably through 2 spin operators. Since we have a 2 spin system here, it can only go up to 2 spins in the simulation. And we compare it against RFDR here, and this slide is a little bit busy with colors, but like just to focus on the blue and red terms, which are the XY terms which build up in RFDR. So during the pulses, there are many other operators which are excited, but basically after the pulses, we see we just have the blue and red operators built up. And over a powder average over the sample, we build up these 2 spin operators as you might expect. But for the MODIS sequence that we've developed, this is all zeros. So what could be the explanation for that? Well, we thought maybe different crystallites are working differently, and this turns out to be the case. So if we take just a specific orientation of the system, then we do see buildup of these 2 spin operators. Meanwhile, if we take a different orientation, then they have the opposite sign. So this is an interesting characteristic of this sequence that it doesn't make sense to do an intermediate powder average and look at 2 spin operators because different crystallites are operating with opposite signs. But of course, the overall transfer from spin 1 to spin 2 is a product of 2 transfers, and these products always end up with the same sign. So here is the second step where we go from the 2 spin operator back to spin 2, and if we multiply these together, then we always get a positive result in the end. So to summarize this part of the talk, we've developed a selective 0 quantum first order recoupling sequence, which we've tested at both fast and ultra fast spinning, so 55 and 100 kilohertz MAS. It has a good transfer band within about 2 ppm for the amide region, and little transfer beyond about 3 ppm. And the sequence depends on offset differences, not a frequency band. We've also tested it both at 600 megahertz and at 1.2 gigahertz. Initially, we thought this might work well for 600, but let's see at higher field, this might result in a bandwidth which is too small, but this ends up being pretty good for both of these fields. Okay, so I wanted to then switch topics a little bit to a diagonal suppression technique, which we're combining with this selective transfer method, but this can also be combined with broadband sequences like RFDR, and that's what's shown here. And so what we do in this sequence is we make use of the so-called orphan spin operators after cross-polarization, which is something many people are using for multiple acquisition type of sequences, but in this case, we use it for a single acquisition, but to suppress the diagonal. So this is the sequence, the simple CP-based sequence for indirect transfer. So we see cross-polarized to protons and then do, in this case, RFDR and cross-polarize back to detect nitrogen frequencies. So here we just insert these 2 black flip pulses to store the nitrogen signal that was not transferred. And with the correct phase cycling for these 2 inner pulses, then we can cancel diagonal signals. And over on the right, you see the performance. It reduces the diagonal by around 50% in this case, although of course it's dependent on multiple factors, including the mixing time that you use and the details of the cross-polarization steps. But importantly, it has no penalty in signal to noise. So most diagonal suppression techniques either have an extended pulse sequences or maybe some additional acquisitions to suppress the diagonal. In this case, there's no penalty on the signal to noise. And it could be combined, for example, with the diagonal suppression technique that Rasmus had published previously. although we haven't done that yet. So we also thought that this sequence might be useful for lower spinning. So we tried it with a CHHC sequence. But in this case, we don't really get diagonal suppression, and that's because under these conditions, there's pretty efficient PAR transfer during the CP steps. And so the remaining signal is not, that we have here, is no longer just on the diagonal. We also see transfer. But so in this case, you could imagine some different combined sequences, for example, CHHC plus PAR in one sequence. This was shown just for a crystalline histidine sample. So then we've also, in the title, I promised some membrane proteins. And so far, I talked basically about model systems and pull sequences. So at the end, just show a few spectra of some of the systems that we're working on. This sample is strictly not a membrane protein, but the sample conditions involve membranes. And this is a non-native state of IDP protein where now we can use a protonated sample and record these kind of contacts. And you can see several of the diagonal peaks that look to be fully suppressed, although we didn't get too many cross peaks from those sites either. So the diagonal suppression is working a little bit variable across the sequence. It tends to work a little bit better for residues that have some exchangeable protons nearby, which is probably just an extra loss term there. So then we've also applied this to a citrate sensing protein. This is a protein over 300 residues in size, and it's a dimer whose function is still, for signal transduction, is still not really clear. And in this case, you can really clearly see the diagonal suppression working, in particular in this region. And we're working through these cross peaks to help understand the structure. We've also then applied this to helical protein, but as you can imagine, the amide-amide contacts for a helical protein are not as structurally revealing as they are in beta sheet membrane proteins. So in this case, we can confirm the helical geometry of the protein, which we knew already in most cases from the chemical shifts. But we do see contacts up to +3 or maybe even +4 I+4 residues in some cases. So we're excited to be introducing these pull sequences and applying them to these different problems, such as looking at aggregated proteins, which are not really possible to solve by AlphaFold. They're too small for things like cryo-EM, and also in confirming structures or detailed structures in membrane proteins. So in the case of M2, this specific hydrogen bond, which was proposed by Tim Cross many years ago, has only been confirmed in solid state NMR samples and in lipid bilayers. And even in the lipid cubic phase samples, this hydrogen bond interaction was not detected. So the protein seems to be fairly sensitive to its environment and important to use these kind of tools. And with that, I would be happy to take any questions.
Thanks, Lauren, for a great talk. So questions for Lauren?
Yeah, I have two questions. First of all, there will be dipole truncation effect unavoidable, right, in this approach?
Yes, we haven't looked too closely at that, but we expect to have dipolar truncation. So it's not that bad though, because of the selective band for amide protons, we're basically searching for the closest proton, okay, which is in the amide frequency region.
Yeah. Right.
But you're right, and we tried it for aromatic systems, and there that is not the case, and it's much harder to observe the longer distances because aromatic systems tend to have other close by aromatic protons.
The second question is, even though in the observation you see peaks among amide proton, however, during the mixing, the proton magnetization go to everywhere, even to the aliphatic proton, to the water, and then they don't come back, right?
Not too much. So for the most part, this is on the 1D spectrum here. You can make out the amount of signal that ends up on the aliphatic region. And remember that the aliphatic region has many more protons in it. So if it were equally balanced, we'd expect to have much more signal in this aliphatic region. So we do see a little bit of transfer to the aliphatic region, but not too much. Transfer to water should also be suppressed because it has a significant frequency separation, unless it's transferred by exchange, of course. But we haven't looked too closely at the water signal, but if we did have a lot of water signal building up, we should see it in this sequence as well.
One more question. So you use this scheme to, detect only amide proton, but can it be combined with a DAR type of mixing so that it improves, it obtains, the immune to the dipole truncation effect somehow?
I'm not sure exactly what you mean by combining with DAR. So this is a proton-proton transfer step. So maybe you mean a third spin like. Well, I mean, it's a proton-proton recoupling sequence. So we do have a specific set of pulses on proton. I'm not, maybe we can discuss after because I'm not sure how we would combine DAR with this. Especially when we have the diagonal suppression, then we're using the nitrogen path for diagonal suppression, and the proton path is just a pure proton-proton mixing step. So, sorry, I can't hear. To also apply a pulse on the nitrogen.
You can do the type of mixing, and then the mixing between proton-proton, but you can apply, the chop pulse along nitrogen to cover.
Yeah, sure. I think, yeah, we could combine it in the sense that you could sequentially do some amount of transfer among the protons and then some with a third spin assisted kind of recoupling. Maybe not really a DAR, because I mean, DAR is specifically more like applicable up to about 20 kilohertz, and this is at 55 to 100 kilohertz, but maybe a third spin type sequence could be used. Yeah.
Okay, we have another question.
Hello, you gave a really good talk. I was just wondering, when you developed the modest pulse sequence with simulation, did you use something like spin evolution for that?
We did. Although the postdoc has written his own software. So it's run in MATLAB, but essentially it's the same kind of technique. Yeah,
very cool. Thank you.
With exact numerical method. Yeah.
Thank you.
I had a question as well. So you mentioned for the modest that it works 600 to 1.2, hopefully higher field. Could you run it at lower field, or is there, you just wouldn't typically, but could you do it, or is there something fundamental about that?
The only fundamental thing is that we don't have a magnet that's below 600.
Okay, so it wasn't a fundamental limitation of the theory.
Yeah, so I don't know. I can't tell you. Yeah.
All right, thanks. Any further questions? Then join me in thanking Lauren. So a quick announcement before we change speakers. If you are a speaker and you're waiting for a speaker check, please do pick that up from Jennifer at the booth right outside today, or the organizing committee will thank you for the donation. So, okay, our next speaker is Rasmus Linzer. The title of his presentation is Higher Dimensionality Fast MAS Solid State NMR, an unexpectedly viable weapon against peak overlap. Rasmus?
Okay, sorry, I wasn't aware. Okay, now better. Okay, perfect. Good. So right into science. So obviously, dimensions have been able, like added dimensions have been able to disperse signals into higher dispersion and finally enable, for example, unambiguous assignments, unambiguous characterization of peaks or of resonances for multiple decades. And basically, we've been reaching this 4D limit before the development of NUS sampling. And then I was not aware of how to put a 4D picture there. That's why this is not showing anything. And so then with NUS, then even 5D, 60 and 70 spectra have been used, in fact, for IDPs mostly. And all of this was possible because people have been overcoming issues, bottlenecks in labeling and sampling. As I just said, the NUS. However, sensitivity is something that still remains a fundamental bottleneck of all of this. And this is particularly true for solution NMR, where really those targets that require this high dispersion, the big ones, the high molecular weight targets, in particular the globular proteins, they have these very long, I mean, the tau C is long, and so the coherence lifetimes are short, which basically reduces the signal to noise in evolution periods and losses in in-app transfers, which makes these sequences difficult to apply. And so Lauren already gave an introduction to proton detected fast solid NMR, and I just briefly point out three things here that are most important for us. And in fact, we've been working also with deuterated samples that are 100% back exchanged at the amide positions. And so as we spin fast and we don't have any deuterons, any proton dipolar couplings, or strong ones at least, we do not need any high power decoupling. And we can also use very fast cycling. So 3 scans per second is something that's easily achievable with paramagnetic doping. And then the other big issue is that really we have very high transfer efficiencies in T1, like in all of the magnetization transfers. And that is something that allows for high complexity pulse sequences, where one element is added to another one, and a third, and a fourth. We still have a reasonable signal to noise. And the important part is that this really is the same thing for high molecular weight targets, just as much as small molecular weight targets, because they spin with the same rotor period in the rotor. And so all the transfer efficiencies and the T2s usually are very much the same. They are identical, in theory. So that is something that has been put forward by multiple groups, for example, by us, by also Guido Pintacuda, and by us again in different flavors. So all of this just says that basically the R2s are the same, irrespective of molecular weight for solids, and well, in solution we have drastic disadvantage when we go to higher molecular weight targets. So that is a really important advantage of solid state NMR, and in particular higher dimensionality or higher complexity pulse sequences. And so 4D solid state NMR has been introduced a long time ago. So this is an early example by Chad Greenstra's group, but then we also actually work that's closely related to what Lauren just talked to us about, the RFDR or whatever kind of mixing schemes that enable a transfer between one amide group and another amide group. In this case, also diagonal compensated or time shared experiments. Similar work was also done by Beat Meyer's group, using in this case DREAM transfer between methyls. Then there's lots and lots of 4D backbone experiments that us and others came up with. So this is, for example, work regarding HNC-OCA type experiments and HNC-ACO type experiments in the solid state. This is also work by Paul Schander, roughly at the same time. Then we had this beautiful experiment in a 4D flavor, HNC-OCA and H, which is an amide to amide correlation. Also something that as a 3D was introduced at the very same time by Lauren and by our group in 2015. And so this is only the 4D version of it. And then there's a 4D HCCH by Teresa Calomagno here in the ribose, and also 4D sidechain to backbone experiments, which are basically correlating all of the sidechain carbons in this case with the C alpha, the N, and the proton, something like a CCCONH toxi in solution without the CO though. So the 4D is kind of established. And I just wanted to go into more detail for this sequence here as a kind of introductory slide, an added one. And so in contrast to these more established sequences for backbone assignments, which are making use of the carbon shifts to actually link one residue and the preceding one or the successive one, which are based on solution NMR like HNCO plus HNCACO, HNCA plus HNCOCA, HNCACB plus HNCOCACB. That in comparison to these ones, which work for small molecules, for big molecules, really we have lots and lots of overlap, even if we have matchmaking using all three of these types of carbon nuclei, CO, CB, and CO at the same time, we still only get 30% of the residues that are linked unambiguously. And so then this experimental scheme is really a big advantage where one amide is directly linked to the next amide. And as I said, this has come in multiple flavors, in 3D versions and 4D versions, and also in EPSI kind of experiments and very sophisticated other experiments that use multiple pathways at the same time by Jan Stanek, also in Guido's group. And so this is something that really keeps a very high signal to noise of, despite the multiple transfers of around 8% compared to an HSQC when looking at the bulk signal. So that is good, but still, if we have very drastic overlap, that is a problem because the sequential linking, even though we have crossed out the carbon and ambiguity, we still retain the ambiguity due to amide overlap. And so the question, and on the other thing is that also we want to associate some kind of backbone, some kind of residue type information with this sequential linking. So HNC-ACB experiments are usually very crucial for assignments. And again, we have this HN dispersion only for linking these sequential connectivities with the sidechain type information. So the question is, can we, how can we improve this? Sorry. And one way is to actually increase the dimensionality of these experiments and add another carbon C alpha chemical shift evolution. And luckily 5D NMR experiments have been worked out, or the theory for, in particular, the data processing, reconstruction of 5D NUS data has been worked out by Viktor Koszminski's group and others. And we've been interacting with Viktor about this quite fruitfully. And so basically the thing is that as otherwise the data would be terabytes and terabytes, one is only looking at 2D planes out of this 5D cube, which are generated at the coordinates of three, let's say, basic shifts. So we would use a 3D base experiment like an HNCA to just look at those specific H, CA, and N positions to look at, for example, the next residues H and N 2D plane. So that's something that works very well and then gives us the possibility to really link this HNCA type of three-dimensional shift identification or residue identification with C beta or even the whole set of sidechain carbons, which then give this sequential link also the information about what sidechain or what residue type we're looking into. So this is something that increases the level of fidelity. It comes in two flavors. It can be going backwards or forward. They have more or less the same signal to noise compared to the bulk, like 8% or 5%. So the backwards walk is a little bit less sensitive, but in general, it's kind of comparable. And this is just some benchmark experiments. So this is recorded in, within 1.5 days on the small protein SH3 domain, which also Lauren just mentioned. And we get already beautiful spectra here, looking at a 2D HN plane that gives us the chemical shifts of T24 at the H, C, and N positions of the preceding V23. And in this case, we have the backwards walk where we look at T24 shifts H, N, and C alpha, and we get the amide N and the amide H of the preceding V23. And if we apply something like this to a big protein, and this is again work with that we did with a very fruitful collaboration with Lens Group, and in this case, the sample was prepared by Varun Sakrani. This is slightly longer, so this is a 30 days experiment because really this has more than 10-fold more residues. So this is really one of the largest proteins that's out there in solid-state NMR. And you can see here, you get these nice backbone walks from, for example, starting out here at this 84 position, using those H, N, and C alpha chemical shifts as a base. And then you get the next residues, H and N shifts. Or here, looking at the next residues, H, N, and C alpha shifts, we get the then the 86 H and N shifts. So this is something that works very well, and still with this experiment only, we didn't get very far. But if we basically what we did in order to really assign a great extent of tryptophan synthase, we teamed also up with Peter Güntert and incorporating computational assisted assignments using FLIR, which basically makes use of the three-dimensional structure, which is known, and uses shift prediction there, and then really has a lot of combinatorial power to get all of these information processed much more quickly than a human brain can. And so we got to roughly 60% of the assignments that we are that contain H, N, Ca, Cb, and Co that we're fully confident with, and another 90% of the residues where we have part of this group of residue shifts that we are confident with. So this really gave us access to biological questions. Again, something that was very fruitful collaboration with Lens Group, and in particular Jacob Holmes, who's also here in the audience. And so basically, they calculated chemical shifts that would be expected for a certain protonation scenario here in the cofactor pyridoxal phosphate. One such situation and another different situation, similar to what Jacob talked about yesterday, or was it, yeah, some day. And so these are the experimental chemical shifts that we found, and they match exactly the computed chemical shifts when we assume a linear combination of two states with a 60:40 ratio. In fact, that's something that was really beautifully demonstrated also using Bayesian probability statistics. Then we also recorded temperature-dependent data. We recorded line shift, or we did line shift analysis. We looked into dynamics. This is just the crystal structure. B factors, and this is R1 row experiments for the site for the active site of the beta subunit. And all of this together, I don't want to really go into details here due to time, tells us that we have a tautomeric exchange, and this tautomeric exchange has a signature that has a microsecond time scale involved, which is way slower than what you would expect for an isolated tautomeric exchange for a protein that hops between two sites that are so close to each other. So really, this is an event that is not isolated but modulated by the overarching electric or electrostatic conditions that are changing when the protein has some active site conformational exchange. So if we have nicely dispersed HN peaks, then we can get, for example, dispersion in sequential linking with HN-HN40s or even pseudo 40s, like one HNH and one NNH experiment. If we do have some ambiguity in the HN plane, then a real 40 would be better, or as I just said, the 5D is even better because it also gives us the possibility to link this with sidechain type or residue type experiments. However, if we have lots and lots of overlap in the HN plane, what we would ideally want to have is something that identifies one residue with H, N, and C alpha shifts, and also at the same time identifies the next residue with H, N, and C alpha shift, because only then, if the HNCA is resolved, then we get an unambiguous linkage. And so this can be, in fact, done in a sort of pseudo-60 experiment, for example, using HNC and H, HNC and N, and HNC and next residue C again. Or, because that costs the same time, we can also just generate this experiment, and that's then a little bit better, having one 5D experiment, H and C and N and H of the next residue, and then another 4D, which is H and C and the next residue C. So, and that last experiment is something that hasn't existed yet, but becomes possible because, as I said, we have these very long coherence lifetimes and the high transfer efficiencies. And so this is just work in progress, but this is on a small model compound, again, the SH3, not compound protein. And this was recorded in 18 hours, and we get beautiful 4D experiments, which give us, for example, here again, this V23, HN and C alpha shift as a basis, and then we get the next residue C alpha. And so if we combine this with a 5D, that gives us the next residues N and H. Then we have all the next residues. Chemical shifts of the backbone, we can really get very unambiguous connections. So as I said, this is only for SH3 so far, but it has great potential for higher systems. So this is just before Len throws me off the stage, something that I want to touch on. We can also combine sidechain, you can use 5D experiments for getting really unambiguous sidechain assignments. And so a backbone fold can already be obtained pretty nicely on the basis of amide to amide connections. But what we really want is a lot of aliphatics in addition. And this is just a teaser here, nothing that I really talk about. The EFDR project that we've been really working hard on for the last years. And we get these time resolved buildups and lots of different correction factors, and we can finally get very, very high resolution structures. But for all of this, we need aliphatic assignments in the first place. And if we spin at 100 kilohertz and we have fully protonated systems, then the line width of the proton is still so large that really these planes usually look pretty bad. And even if we have 40 HCCH experiments, then we're still stuck with the resolution of the H alpha C alpha to get this connected to the amides. So is there any possibility of getting this done more effectively? And yes, there is the 5D sidechain to backbone experiment where we have the H and the C chemical shifts as kind of a toxi plane that we have that we disperse into H, N, and C alpha chemical shifts in the third, fourth, and fifth dimension. So this is very beautiful data. We can combine this even with a 4D HCCH experiment, and by this really get, so this is kind of a little bit more details that I don't have the time to go into detail for, where we have to project down the 5-dimensional data into 4 dimensions if we record this simultaneously. But we get lots and lots of sidechain aliphatic shifts that we can then use for structure calculation. So with this, I'll conclude, and I hope I convinced you that we got very long coherence lifetimes and short recycle delays, and then as such, complex pulse sequences become very well suited for solid state NMR. And this is also true even for high molecular weight targets, which are the real proteins that require this. And so fast magic angle spinning brings important new prospects for high molecular weight targets. So with this, I would like to thank the people involved and you for your attention, and I'm happy to take any questions.
Thanks, Rasmus, for an absolutely beautiful talk. So we have time for a quick question. Yep.
Why do you want to evolve the amide proton with a short lifetime? Why not NCACO, evolve carbon in the 5D? Yeah,
that's true. Usually we characterize our spin system, like the proton has been, or the amide proton nitrogen correlation has been the fingerprint. So if we have an amide pair that we can relate to, this is very useful. But you're right, if you have a carbon dimension in there, this could potentially bring better dispersion, particularly the proton. It's all possible. So obviously, and for example, in this work by Paul Schander that I mentioned, this HNC-OCA, they kind of rather refer to the carbons rather than the two protons, for example. So you're right.
Okay, well, to keep the program moving along, let's thank Rasmus again. Okay, so our next speaker is Matt Eddy. The title of his presentation is The Structural Basis for Regulation of a Human GPCR by Membrane Lipids Investigated by NMR Spectroscopy. Matt.
Thank you very much. This is a solution NMR talk, but I think will be of also interest to the solid state NMR community. So our group is very interested in GPCRs, or G protein coupled receptors. These are 7 transmembrane proteins on the surfaces of all eukaryotic organisms, pictured here, shown in blue. These respond to a vast range of extracellular molecules, including hormones, neurotransmitters, as well as about 1/3 of FDA-approved drugs. And activating drugs will interact with the receptor and cause a complex formation to occur with intracellular partner proteins that kick off signaling cascades, ultimately leading to changes in cell physiology. So pictured here in the background is almost an afterthought is our cell membrane. Shown here is a kind of representative view of the current state of the literature, showing it to be a very detail-less feature and almost an afterthought. So we're very interested in understanding the influence of this lipid environment on GPCR drug responses, as is poorly understood. The other motivation for this is that the vast range of GPCR structures have been determined in the absence of a lipid environment. So in crystallography, all of these crystal structures are determined using a molecule called monoolin, forms lipidic cubic phase, which is not physiologically relevant. And actually, even via cryo-EM, many of these structures still are determined using detergent micelles. So shown here is this kind of balloon around a GPCR structure. So there's an increasing interest in this in the literature, both in the experimental and computational communities. From the computational community, there have been predictions showing the importance of anionic lipids as molecules that facilitate the formation of these signaling complexes. So shown here is some work from Mark Sampson's group, where we're looking at now a complex of a receptor with a partner protein, shown in blue here. And these red areas that you see here are areas that are predicted to interact specifically with anionic lipids, such as POPS and PIP2. And the prediction is that such lipids are required for formation of competent signaling complexes. So we're studying these, the role of anionic lipids and formation of signaling complexes using a representative human GPCR. This is an adenosine receptor. So this interacts with molecules found in chocolate and caffeine and tea, but it's also important in regulating a number of physiological processes as a validated Parkinson's disease target, and also as a cancer co-immunotherapy target. We have a number of structures of this protein, and so it's also great for developing new kinds of biophysical techniques to get at fundamental questions like the role of lipid bilayers. So what we're using, the model system that we're using here are lipid nanodiscs, a cartoon of which is shown here. These are formed by these so-called membrane scaffold proteins or belt proteins, shown as blue helices that corral together clusters of lipids into which we place our GPCR. We can form very homogeneous preparations of these, shown here, even just using these standard scaffold proteins that have been available for a long time from the Sliger lab. So shown here is a superposition of 2 spectra. So one is TROSY spectrum of this receptor in detergent micelles, that's the red spectrum here. And you see also superimposed on that is a spectrum of the receptor in lipid nanodiscs, shown in blue. The spectra are identical. I'm not going to get into a detailed analysis of this data right now, but just to highlight the fact that the protein is folded within lipid bilayers. So we like using the nanodiscs because we can precisely control the lipid composition within them. And in this particular study, we're looking at mixtures of both zwitterionic lipids like POPC and POPE with anionic lipids like PG, PS, PA, and PIP2, an important signaling molecule. We use phosphorus 31 p-NMR to convince ourselves that the composition within the disk is really what we intend it to be. I'm not going to go through all of the detail here, but just to point out that essentially what you want to put in the disk is what ends up in the disk itself, which was actually surprising to me to see. I'll also point out that we've done systematic measurements of ligand binding of the receptor in lipid nanodiscs using standard radioligand binding assays. And the conclusion from this is that the lipid composition itself has a very minimal impact on the dissociation constants of drugs with this receptor. So that's important to keep in mind for the subsequent NMR data that I'm going to show you. Also, we've done systematic measurements of the protein fold across a range of lipid compositions. We're looking at the results of thermal unfolding assays shown here. As we essentially increase lipid composition, the composition of anionic lipids going from the left to the right curve, we see that this inflection point increases. So the receptor is folded across a range of lipid compositions, but as we increase the proportion of anionic lipids, we see an increase in the melting temperature of the protein. And that's true for a wide range of lipids, as shown here. And in particular, compositions with PS. So PS appears to play a special role in particularly increasing the relative stability of the protein. So we're using fluorine NMR to do these kinds of experiments. This is a very cost-effective and efficient way of screening a wide range of lipid compositions. It's been popularly used by a number of groups in solution and solid-state NMR. We can introduce a specific fluorine NMR probe at this position here on the intracellular surface of the protein. This is chosen specifically because the structure of the protein changes in and around that particular site in response to different kinds of drugs. So we expect to be able to measure that response by NMR. And we can use standard microaddition phylochemistry to attach a very sensitive CF3 probe to this specific position. And that's the only position in the entire protein that ends up being labeled. So here's some representative simple one-dimensional fluorine NMR spectra of what we see for the receptor in detergent micelles. So this top spectrum here is what you see when we add an inactivating or antagonist drug to our receptors. So we have essentially a spectrum with two components in our signal envelope. And in the bottom spectrum, we see a very different signal envelope. And this is because we've just added an activating drug or agonist to our protein. The reason that we see multiple components is because we actually have the presence of multiple simultaneously populated conformers that are observed, the relative populations of which are proportionate to the intensities of the signals that we see. And so when we look at changes in signal intensities, we're looking at changes also in relative populations of the receptor. We can deconvolute these spectra. This is consistent with previously published data from the Vutrich group that I was also involved with. And when we do that, we see we can define these states P1 through P4. And in particular, we have different populations that depend, again, on the efficacy of the drug that's bound to the receptor. And I want to particularly pay attention to this state P4 that you see here on the left side. So what happens if we compare this data to what we get in lipid nanodiscs? So these are discs composed of a mixture of PC and PS. And if we just scroll across the top, we see our NMR signal envelopes are essentially very similar, identical when we look at complexes with inactivating drugs. So same chemical, very similar chemical shifts. But the situation is actually a bit different when we look at complexes with activating drugs. So again, we see these states P1 through P4 with surprisingly highly similar chemical shifts, but very different changes in relative intensities, indicating we have completely different relative populations that are only different because we've changed the environment in which the protein is in. This is also reflected in terms of change of conformational exchange rates. So if we compare here, these are 2D exchange spectroscopy experiments on the left. This is work that was done in detergent micelles where we see cross peaks between two of these states, indicating a rate of exchange faster than 10 per second. And those are completely absent when we go to our nanodisk environment, indicating that we have to have rates of exchange that are at least an order of magnitude slower within lipid bilayers. So now it gets a little bit more interesting when we start to compare different lipid environments, again, using a simple 1D fluorine NMR method. So what happens when we look at samples prepared in just 100% POPC? This is a spectrum of the receptor in complex, again, with the same inactivating drug, essentially the same. But now when we look at a complex with an agonist or activating drug, we're completely missing this state P4 here, which is completely absent now when we look at nanodisk composed of just zwitterionic lipids. So we can assign that state P4 in a kind of functional manner. And we can do that by, again, preparing samples in just nanodisk containing POPC. We can add an activating drug to that. And initially, what we see here is mostly a profile or a spectrum that looks highly similar to the inactive receptor. And to that same sample, we can add a G protein. So that's shown here in blue. And in so doing, we can then populate, again, this state P4 that you see on the left here. So this state P4, we assign to representing a fully active conformation of the receptor. So now we can look at the impact of lipid composition, keeping in mind, again, the assignments that we just saw. So we have in the top spectrum here, nanodisk composed of just POPC, so it's shown here. And now if we look at subsequent spectra where we add in different anionic lipids, such as POPS, POPA, PG, and PIP2, we again see the presence of this state P4 that was completely absent when we had a lack of anionic lipids. So from this, we can conclude that anionic lipids are required to populate a fully active conformation of the receptor. So this generated the hypothesis that it was the presence of charged amino acids that were near the predicted bilayer boundary that could facilitate or mediate these specific interactions. And in particular, we focused on a cluster of charged or amino acids that are predicted to be charged, that are also predicted to occur near the lipid bilayer boundary. So these here, 4 that we're focusing on, 2 of which are highly conserved among hundreds of different GPCRs. So this lysine and histidine, for example, are observed not only in class A receptors, which are rhodopsin-like receptors, but also among all GPCRs, including adhesion receptors, glucagon, and other receptors that are not directly physiologically related to class A receptors. So what happens if we introduce mutations to these proteins? Introduce mutations here. So in the top, what we are looking at is the fluorine NMR spectra for this initial protein that we worked with. Again, where we see complexes with activating drugs, we only see the formation of this active state when we add anionic lipids shown here. So when we introduce mutations, so this is an H230Q mutation, we essentially invert what we had just seen. And so in the absence of any anionic lipids, we see the presence of this state P4. And then in the presence of anionic lipids, it's completely absent, shown here on the right. We see a similar observation if we introduce this lysine 233Q mutation. So again, in the absence of anionic lipids, we see the formation of the state P4. In the presence of anionic lipids, we see a very, very small population of this state, but obviously highly diminished. And now if we look at, as a kind of control, if we look at a mutation in an adjacent residue, sorry, in an adjacent helix, this is an arginine to 291 Q mutation, we can essentially recover this initial spectrum that we had seen here. So these two mutations in the middle are present in helix 6 of this protein. And this helix undergoes the largest conformational changes in the entire protein upon activation and formation of signaling complexes. Whereas this mutation in helix 7 is in a location that doesn't undergo such large conformational changes. So this led us to speculate a potential mechanism for what's going on here that was inspired by work by Tobias Almer back in 2012, and that it was the presence of actually these charged amino acids that are near the lipid bilayer interface with the intracellular solvent that mediated a kind of snorkeling effect in helix 6. So this is just a current hypothesis of what might be going on. But if it's true, it would be a completely unique idea for understanding GPCR activation and would be in some ways a very universal observation applicable to potentially hundreds of GPCRs. So just to kind of sum up a little bit of this work, we see that anionic lipids are required to populate an active conformation of the receptor. There's a synergy that occurs between drug efficacy and the lipid environment. And so it's not just the efficacy of the drug, meaning whether you have an inactive or activating drug present that is important, which has been the focus of most previous work, but also the lipid environment itself appears to heavily modify the activity of drugs. Positively charged amino acids present at the surface appear to mediate these interactions. And I'll just also point out that lipid composition makes a huge difference in biophysical and structural studies. So looking at the literature, I get the impression that lipid nanodiscs are often used in a way of sort of whatever works. That's what we're going to use. And there's not necessarily a goal to create the most physiologically relevant environment within lipid nanodiscs. And I think that moving forward, that this needs to be paid very closely attention. It needs to be paid closely to. So with that, I want to acknowledge this work, which was done by several graduate students, several of which are here, and our rapidly growing group. We're very much interested in hiring postdocs and future graduate students. So if this is work that interests you, please come and talk with me. And I'll just also point out that this is actually a very simplistic, fluorine NMR is a very simple method, but very broad tool for studying effects of membrane composition. So this is work also done by ARCA, presented at this meeting, looking at the impacts of cholesterol and how the presence of other lipids also modulate the response of cholesterol to the receptor. So with that, I'd be happy to take any questions that you might have.
Thanks, Matt, for a really great presentation. So questions?
Really nice talk. I have a question about the POPC experiments you did on wild type. So when you showed that even though the agonist doesn't trigger the P4 conformation, the G protein does trigger the, can you go back to that slide? Sorry.
Yeah, so what you're looking at here is, that's right. So we can kind of force the formation of this signaling complex. So what's present in this sample is the receptor in POPC with a saturating amount of an agonist, and then to that we add this G protein, and we can force this, essentially force a percentage of the complex to form. This peak height, this peak intensity though, is much lower than what you would see if you do the same experiment and you have POPS also present there. So
yeah, so my question is that in the top spectra, doesn't look like you have any creation of P4 in in that system, but you do make it in the bottom one. So my question is, if you don't add the agonist, but you add the G protein, do you still see the P4 conformation? And what do you Yeah, did you? try
No,
that?
so you don't, so one, the basal activity of this protein is very low. So the APO receptor, or the data with the APO receptor on its own look like you have an inactive receptor antagonist-bound protein. And if you add the mini-G here, you don't really see much of an increase in that population either. So it also looks like a kind of inactive receptor.
So you don't see any P4 with just the G protein.
Right,
Yeah.
right, yeah.
Thanks.
And that may depend, that may be specific to this receptor also because of its low basal activity.
We have another question.
Thanks a lot for this very clear talk, actually. I was wondering exactly about when you do your peak deconvolution, we see apparently different line widths and probably different dynamics in these individual states. It's kind of twofold question. So do you understand what the dynamics are, and do the lipids have an influence on the dynamics?
Yeah, so well, one, yes, the lipids do have a large influence on the dynamics. So I mean, we have done at least these exchange spectroscopy experiments where we see, in comparison between previous data that was published for the detergent solubilized sample, the exchange rates are slower. We don't know exactly how much slower, but they have to be at least an order of magnitude slower. The other thing is some of the previous work that was done was just using saturation transfer experiments, where there we could quantify those rates of exchange between these different states. And just to sum up that work, they're all on all of these are slow exchange processes. So they're on the order of 10 per second to 1 per second, but very, very slow. And that's also consistent with other fluorine NMR data that was done for other GPCRs as well, that most of these, so these different conformations and their exchange rates are typically very slow. So the idea is that this must correspond to large conformational changes in the protein.
Thanks a lot.
Okay, if there are no further questions, let's think about it again. Our next speaker is Blake Wilson. The title of his presentation is Time Resolved DNP Enhanced Solid State NMR Captures Protein Folding and Oligomerization Triggered by Sub-millisecond Negative Temperature Jumps. Blake?
All right. Thank you. Good morning, everyone. So my talk will be a little change of pace from the previous talks in this session. I'll be talking about relatively low field NMR, 400 megahertz NMR experiments with 4mm rotors and relatively slow spinning speeds, and really be emphasizing the methods development and the instrumentation work that we're doing to address fundamental biophysical questions. So many biological systems, like globular proteins and protein assemblies, have the property that they form well-ordered states at physiological temperatures, but unfold and become disordered when you heat them to high temperatures. So of course, we all know that temperature-induced changes are a classic way of studying protein folding and energetics. The experiments that I'll be talking about today are based around the following relatively simple idea. So what we do is we heat a system of proteins to high temperature to cause them to unfold and become disordered. For example, to 95C, as shown here. And then we rapidly cool the protein solution to below wherever the folding transition is, in this case, say to 30 degrees C, to induce folding. And the goal is to carry out these rapid temperature drops on a timescale that's fast compared to this kind of timescale of protein folding. And if we can do this, then we can carry out something like a native folding experiment from the thermally denatured state to the thermally folded state under something like physiological conditions. So small proteins and protein assemblies can fold very rapidly on the sub-millisecond timescale. And that sets the limit on how quickly we should be able to change temperatures if we want to capture interesting dynamics. And furthermore, if we want to get structural information about any intermediates, as well as kinetic information, then we need a way to probe the system with a time resolution that's also on the millisecond timescale. So today I'll tell you about how we do these experiments and show you some of the results that we can get with these techniques. So we figured out how to carry out rapid temperature drops using a flow apparatus, which I'm showing schematically here. We use an HPLC pump to flow a solution containing proteins through a 100 micron diameter copper capillary, which we solder to a heated copper plate to raise the temperature of the solution, in this case to 95 degrees C. We then flow the solution through a second narrower copper capillary with a 30 micron inner diameter, which is soldered to a second copper plate, which we cool with ice water. This drops the temperature from 95 C to 30 C in about 0.8 milliseconds, so extremely rapidly, using the 1 milliliter per minute flow rate, which is sort of the typical flow rate that we use in these experiments. And then the solution exits the 30 micron capillary as a high velocity jet. Our strategy to study the evolution of the system is to freeze trap the protein solution after the temperature jump, capturing frozen snapshots of different time points in the evolution process. And we do this by injecting the high velocity jet directly into a cold isopentane bath, which we cool to minus 145 C using liquid nitrogen. And this technique has been shown in previous studies to rapidly freeze solutions of protein that are injected into the liquid. The jet breaks up into small particles that freeze on the timescale of 10 to 100 microseconds in the isopentane solution. And then by varying the distance over which the jet travels or the flow rates, we can probe different time points after the temperature drop to get kinetic information about the folding process. So isopentane provides very rapid freezing, but then it becomes a bit of a challenge to get your sample out of the isopentane once you've done this experiment. So the protocol we developed to do that, I'm briefly outlining here. We filter the isopentane using a fine mesh to remove the frozen ice particles, which we then store in liquid nitrogen until we're ready to pack them into an NMR rotor. And then we do everything at or below around minus 160 degrees C, which is the melting point of isopentane. We use pre-cooled rotors and pre-cooled packing tools to pack the frozen particles into a rotor, which we then store in liquid nitrogen until we're ready to do experiments. And the experiments that we do are low temperature, solid state NMR spectroscopy with magic angle spinning at 400 megahertz with the typical spinning speed of around 7 kilohertz. So NMR provides residue-specific quantitative information, which is great for accessing these intermediate states. But of course, our measurements require cryogenic temperatures because we have to keep these precious freeze-trapped proteins cold. So we have a challenge, which is that we have weak signals because NMR is an intrinsically insensitive technique, from broad lines because we're working at low temperatures and our samples are highly disordered. And our concentrations are on the millimolar or less level for these proteins. So how we address this is we selectively isotope label only certain residues. That's very important. Otherwise, our spectra become much too crowded. And second, we use dynamic nuclear polarization to boost the sensitivity of our experiment. So briefly, we carry out MAS-DNP using a home-built setup, which I'm showing here. We operate at 25 Kelvin. So we operate at very low temperatures using helium cooling, which cools only the central portion of the rotor, while we use room temperature nitrogen gas to spin the rotor with the drive and bearing gas. And in this way, we can maintain low sample temperatures while using relatively little helium. Our helium consumption rate is around 1.5 liters per hour at 25 Kelvin, which would be much higher if we were also spinning with helium. And then for DNP, we're using a 1.5 watt EIO source operating at 263 gigahertz. And crucial for our experiments, we can change samples while keeping everything cold. So that's just a matter of dropping the probe below the magnet, swapping out your sample from liquid nitrogen storage, and then putting the probe back in the magnet, which takes about 4 minutes, 4 to 5 minutes, and is relatively straightforward if you've done it once or twice. And we flow helium the whole time to keep everything cold. So operating at 25 Kelvin offers a couple of big advantages. One advantage is we need less microwave power to achieve efficient DNP, but the really big advantage is we also have, on top of the enhancement from DNP, we have a larger Boltzmann polarization, so our signals are very large. So today I'll talk about 2 applications of our rapid temperature jump technique. I'm first going to talk about how we can use thermal folding and unfolding experiments to study folding and oligomerization in melatonin. And then I'll talk about applications of rapid negative temperature jumps, rapid cooling experiments to study ultra-fast folding proteins. So melatonin is a 26 residue peptide which is found in bee venom. Melittin forms alpha helical tetramers, shown here in the center of the slide, at room temperature and at neutral pH, but becomes disordered when you heat it to high temperature or drop it to low pH. So at neutral pH and at 0.7 millimolar, which is the condition that I used in the experiments I'm showing here, melittin is largely helical at 30 degrees C, but unfolds as you heat the protein to higher temperatures, as shown in the CD spectrum here. We synthesize our melittin in a peptide synthesizer, which gives us an easy way to attach isotope labels to specific residues, which I'm showing here, which were located at points of intermolecular contact in the crystal structure. So here are DNP enhanced NMR spectra recorded on the melittin samples that were prepared at 95 C and frozen on the top and at 30 C and frozen on the bottom. And these spectra were required by first polarizing proton spins with DNP, then cross-polarizing to carbon spins, doing double quantum filtering, and then detecting. And we can see that we're capturing a temperature-dependent conformation distribution in these spectra. In particular, we observe frequency shifts in the alpha carbon positions at the 3 labeled residues that are consistent with a largely random coil population at elevated temperatures and a more ordered helical population at 30 C. And moreover, we just noticed that the lines are considerably broader in the spectrum frozen from high temperature, indicating a large amount of conformational disorder, which you'd expect in an unfolded protein. However, briefly, there's a challenge. I don't want to spend too much time on this, but the isopentane, you can never get all of it out of your sample. So there's always a little isopentane left. At 25 Kelvin, where isopentane is a solid, we see a strong CP signal from natural abundance carbon and isopentane. We can filter that with double quantum filtering, as I showed on the last slide, or we realize we can filter it using simple saturation recovery and inversion recovery techniques, since the buildup of the isopentane signal, which is not DNP enhanced, because the isopentane and the frozen peptide solution containing our radicals are phase separated. Since the signals build up with different times, we can eliminate the isopentane signal by introducing an inversion pulse on the proton channel before we carry out CP, and then do all of our experiments at the null point of the isopentane signal and remove the background isopentane signal efficiently. So just to show you what that looks like, on the left is a 2D spectrum acquired with DNP without the inversion recovery filter, and on the right is a spectrum acquired with the inversion recovery filter. So the baselines are cleaner, and we remove a significant amount of a noise that's present in the indirect dimension with the isopentane signal. So here are carbon-carbon 2D spectra acquired on samples frozen from 95C and frozen from 30C. And we can see, just as in the 1D case, that there's a clear shift in the alpha carbon resonances and an overall line narrowing and from the lower temperature spectra. So the question that we asked is, how does this folding happen? How does the protein fold if it's cooled quickly from high temperature? What are the intermediates in this process? In particular, does the helix formation precede oligomerization, or do helices form concurrently with oligomerization? So previous work with rapid pH jump experiments showed that after a pH jump, helix formation occurs concurrently with oligomerization. We wanted to see if the same thing happened after a rapid temperature jump. So to answer this, we used our temperature jump apparatus to rapidly cool and then refreeze melittin after a short delay. So as you can see here, as we increase the evolution time after the temperature drop, we see an overall narrowing in the line shape. And we also see that the alpha carbon frequencies shift from this predominantly random coil configuration to a more ordered configuration after a long evolution time. This is even more clear in the 2D spectrum again, where here we're seeing pronounced shifts and line narrowing as the evolution continues. So to quantify the timescales of helix formation and oligomerization, we looked at the buildup of intermolecular cross peaks. So here on the top, I'm showing a 2D spectrum acquired with a short spin diffusion time. So we're only seeing intra-residue cross peaks. And on the bottom, I'm showing a spectrum acquired with a 1 second spin diffusion time. So we see these inter-residue cross peaks as well as intra-residue cross peaks. And by looking at the buildup of cross peaks between neighboring residues in the helical state, we can observe the timescale over which melatonin forms a helical configuration. And by looking at the buildup of cross peaks between sites that are only neighbors in the oligomer state, looks like the projector's gone down. All right, it looks like we're getting a new projector. Excellent. Thank you so much. So as I was saying, by looking at the buildup of these two sets of intermolecular or interresidue cross peaks, rather, we can look at the buildup of oligomerization and helicity separately. So we see that these occur on a similar time scale. That was the first punchline of my talk. And since alpha helices can form very quickly in small proteins on the millisecond or even the microsecond time scale, but this buildup is taking several milliseconds, then we can conclude that the helix formation is probably stabilized by oligomerization, by contacts between molecules in the oligomer. And we can rationalize this by looking at the crystal structure and seeing that the tetramer forms a hydrophobic core, which can help shield the helix formation and stabilize the helical conformation. We were able to model the buildup of both the helix formation and oligomerization using a simple unidirectional kinetic model, where we assume that the rate limiting step is dimerization. And if we look at the buildup of the two cross peaks, we extract identical effective rate constants for these two processes. So we think that we have some idea about what's going on here. So I just really quickly, in the last minute or two here, I hope I have another minute or two. I'd like to briefly tell you about a second application of negative temperature jumps, which is to study ultra-fast folding proteins. So the 35 residue Dillon headpiece subdomain HB35 is one of the fastest folding proteins known. Experiments shown here on the right were carried out using a temperature jump, using a high intensity laser pulse to rapidly heat a small volume of sample. And these experiments like this have shown that HB35 folds on the microsecond or few microsecond timescale for the mutant that or for the wild type, which I'm looking at. And then there are certain mutants that fold even faster. And the signature here is the partial quenching of the tryptophan fluorescence, which is recorded as a function of time after this high intensity laser pulse. So a number of years ago, our lab, Rob's lab, investigated thermal denaturation in in HB35 using solid state NMR. So in these experiments, a water glycerol solution containing HB35 was heated to 90 degrees C and then rapidly frozen in liquid isopentane. And on the bottom is a 2D carbon-carbon spectra acquired on this sample. And we can compare that to a spectrum acquired on the top, which was acquired on a slow frozen sample. And by that, I mean the water glycerol solution containing the peptide was put in the rotor was dunked in liquid nitrogen, and the rotor froze on a few second timescale, 5 seconds or so. So when the solution cools slowly, the fast folding proteins remain more or less at equilibrium with the solution. And the final folded state is the energy minimizing structure. 2 residues were carbon-13 labeled in these experiments. And what I want to draw your attention to is valine 50, highlighted here on the right, which in the folded configuration adopts this particular configuration characterized by a splitting in the gamma cross peaks, which is illustrated here in the C alpha to C gamma and C beta to C gamma cross peaks. In the rapidly frozen spectrum, this splitting was not observed. And in addition, there was a downfield shift in both of the alpha carbon resonances consistent with a small population of strongly unfolded proteins. Since no negative temperature jumps were carried out here, no evolution time, the conclusion from these studies was that folding must occur at least as slowly as the cooling time, which is estimated here to be about 10 to 20 microseconds under these conditions. So we recently began investigating thermal refolding of HB35 using our temperature jump apparatus. So here are DNP enhanced carbon-13 detected spectra using double quantum filtering of slowly frozen HB35 and HB35 rapidly frozen from 30C. The 2 spectra are qualitatively quite similar, although the slowly frozen sample overall shows narrower line widths, but we can compare that to spectra that were acquired after a rapid negative temperature jump. We see that the splitting of the valine 50 methyl carbon peaks is absent after a 1.5 millisecond evolution time after this temperature drop, and even after a 3 millisecond evolution time, it hasn't fully developed. So these results are quite preliminary, but we can infer some interesting facts from this. So the overall similarity of the line shapes, and in particular the alpha carbon positions, are consistent with a model where the secondary structure forms very quickly, but apparently full structural order, including the ordering of the side chains, takes much longer, perhaps on the millisecond timescale or so. So we'll have a clearer understanding of these questions as we proceed with these experiments. So we believe rapid negative temperature jumps are a promising technique for studying biological dynamics and kinetics in a variety of systems. We hope to keep doing these experiments, and we'd love to get feedback from people here about interesting systems to study. So I'd like to thank Rob, my boss, Dr. Ming, Dr. Jaekyun Jeon, and Dr. Kent Thurber for all their help carrying out these experiments, and also for the rest of the TIGO lab. And I'd like to thank the organizers for setting up this wonderful conference and giving me this opportunity. And thank you all for listening here on Friday morning.
Thanks, Blake, for a really interesting talk. We have a question.
Yeah, great talk. So I would like to ask about the presence of the glycerol. So without glycerol, perhaps you cannot make the sample in glass state. So you mix it for DNP,
Yes, that's
measurement.
Yes, that's important. Yes.
And what is the sample concentration?
So all of the fast frozen samples that I showed, we were using a 20% glycerol sample. You make a good point that lowering the glycerol concentration below that, you worry that maybe you're not forming a vitreous ice, even if you cool very rapidly.
Okay. Do you mix also D2O in the solver?
Yes, yes, we do. Yes. So these solvents were largely deuterated. There's a 10% protonated solvent, 20% deuterated glycerol, and the rest is D2O and buffer.
Okay, thanks.
And a second question.
Very nice talk. I have a question about the VT MIS setup. So are you afraid of nitrogen condensation? Because you have a very cold and second, do you have an idea about the gradients, the temperature gradients that you can have in your sample, and if this is a problem?
Yes, that's a very good point. So nitrogen condensation and nitrogen freezing in practice hasn't been a huge problem. The only time where you run into this is when you have to stop spinning when you're changing samples. Sometimes a small amount of nitrogen ice can form, but it's usually possible to blow that nitrogen ice away by just slowing the helium flow briefly for a few seconds, or reducing it at least, and then turning it back on. We purge pretty vigorously the area around the NMR stator with dry nitrogen gas. Your second question was about temperature gradients. So Certainly there exist temperature gradients across the rotor. In the sample region itself, at least when operating at 25K, the temperature gradients are not, well, the whole sample is kept very cold and we see strong enhancement from all parts of the sample. The ends of the rotor have Teflon spacers in them, and those are the regions of the rotor that are near the drive and bearing gas. So those are the regions where we would see the largest temperature gradients.
Great, thanks.
Next question.
Hi, very nice talk. I was wondering, these temperature jump apparatus, how do you assess the time scale of the temperature jump? I know you said something when you go from 95 to 30, you said the number, I forgot. Like, how do you measure this? And then the same question applies, I guess, for the freezing and the isopentane.
Yeah, great. So measuring the temperature jump, we characterize first what kinds of temperature jumps we can achieve in these systems with copper capillaries soldered to copper plates. The time is really the average time. It's the time that it takes the solution to flow through these channels at a particular flow rate. We establish something like plug flow. We have upstream a small amount of hydrocarbon to separate the protein solution from the driving liquid that we're pushing out of the HPLC pump. So we achieve something like plug flow, not exactly plug flow, I don't want to say, but the time is really the average time, I should say, achieved by flowing through these regions. So that's just the flow rate and the length of the tube and the area, the cross-sectional area of the tube. And your second question was about freezing and isopentane. So we estimated the freezing time by looking at the size of the particles and the distribution of particle sizes, and then just carrying out simple heat transfer equations and assuming water glycerol glasses aren't perturbed too much by the presence of the protein. And we're looking at the time in particular that it takes the protein to freeze below, below 0 C or below 10 minus 20 C, minus 50 C, something like that, where we expect the folding rates to then become negligibly slow for the rest of the experiment.
All right, cool, thanks.
And one final question.
Good, a quick question, very good talk. When you have a mixture of the isopentane, is it within your sample? So that means, is it then part of the DNP process?
There is some isopentane in the sample, but the isopentane and the water glycerol mixture are not miscible. So they're totally phase separated from each other.
Okay, so it's just a separate phase that you have in your rotor.
Yes.
Thank you.
Well, great. Thanks again for a wonderful presentation. Okay, our final speaker of this session is Nate Traceth, and the title of his presentation is Molecular Mechanisms of Multi-Drug Efflux Pumps. Nate?
Right. Thank you very much for the invitation and for the opportunity to tell you about what we've been working on. So I'll start with an acknowledgement slide first because I may go over by a minute or two. The people that were involved in the two projects that I'll hopefully be able to tell you about, both of which are membrane protein efflux pumps, are EMRE, which was done by a very talented postdoc, John Ping Lee, and a talented graduate student, Callie Saher. And the NORE project, which is a Staphylococcus aureus multi-drug efflux pump, was largely the PhD work of Doug Browley, who was a jointly mentored student in my group and in Donning Long's group. So the topic for today is antibiotic resistance, which you hopefully are already aware is a significant global problem. It affects every place on Earth. There are more than 1 million deaths attributed to antimicrobial resistance each year. And this is predicted to become an even bigger problem with time. And the basic idea is that our antibiotics don't work as well as they used to work because of resistant mechanisms. And to highlight this idea of the prevalence of drug resistance, this is a world map showing you the frequency of Staphylococcus aureus isolates that display drug resistance. So you can see it's sort of prevalent throughout the world and it's a significant problem. So fortunately for bacteria, they have a number of mechanisms to give resistance to our antibiotics. That's unfortunate for us as citizens of the world. Several mechanisms enable the organism to confer resistance to our antibiotics. The mechanism that my group has focused on for about 10 years now is the mechanism through membrane protein efflux pumps. And these efflux pumps are able to do something fairly simple. They pump out, they transport antibiotics from the cytoplasm located in here out of the cell. And in the process, they lower the intracellular concentration of the antibiotic and therefore allow the organism to survive in a higher amount of antibiotic concentration outside the cell. So these are membrane proteins that carry out transport of drugs. 4 out of the 5 major families of efflux pumps perform a fairly simple reaction across a membrane, which is to efflux drugs out of the cell, and that energy source is provided by a proton gradient. So both a chemical and electrical potential proton motive force of that proton being pumped. And in these antiporters, the key residues, the catalytic residues in just about every one of them are aspartate and glutamate residues. So residues that can bind and release protons that can undergo acid-base chemistry. So they can bind to protons through these glutamate aspartate residues. They can release them on the other side of the membrane. This process is called alternating exchange mechanism, where they can move some substrate across the membrane. They can also undergo deprotonation and bind different substrates. And so the topics for today are to try to understand, provide some insight into the mechanism of proton coupling for this E. coli E-Marie protein. And then if I have time, I'll tell you a little bit about how we've targeted those anionic residues in a Staphylococcus aureus transporter called NOR-A. So a little bit of background about E-Marie, which again is an E. coli multi-drug transporter. E-Marie contains all the essential features that the larger transporters consist of. E-Marie is a 110 residue protein. Its key residue that was identified by Shimon Schildener several years ago is glutamate 14. And you can do some simple microbiological experiments where you encode the E-Marie gene on a plasmid, you introduce that into E. coli, and then look at its growth phenotype as you dilute it relative to controls. Now, this glutamate residue is highly conserved. Even if you change it to an aspartate residue, as you can see here, E14D, that doesn't give resistance to this Ethidium molecule. So I should mention that E-Marie is a model system. It gives resistance to mostly antiseptic molecules. There's also some very beautiful structural work that's come out recently. There's some very nice NMR work from Mei Hung's group in collaboration with Katie Hensler-Wildman's lab that solved structures of tetraphenylphosphonium bound molecules at 2 different pH values. And then there's also crystal structure, high resolution crystal structure that came out of Randy Stockbridge's group, and that's what's displayed here. Now, E-Marie needs to function as a dimer. So its oligomeric structure is a dimer. That's the functional unit for transporting drugs, for effluxing drugs. Now, E-Marie is kind of a peculiar system in that the 2 monomers in the dimer are oriented anti-parallel with respect to one another. So they have a feature called dual topology that Gunnar von Hein identified several years ago. So one of the key questions then was to try to understand what the role of this key catalytic residue in this transporter, which is this glutamate 14 residue. So we envisioned that a useful approach to investigating this the specific protonation state of the glutamate residue would be to use solid state NMR, and namely a double quantum, single quantum experiment. Due to the fact that the glutamate and the aspartate residues are quite frequency selective, or located in a very frequency selective region of the spectrum in this double quantum, single quantum correlation relative to the backbone site. So we could get some natural frequency differences to get resolution. These 2 sites also overlap, however, with glutamine and asparagine. As you know, the difference between the asparagine and the glutamine is simply this NH2. So we could apply read or dephasing pulses at the end of this double quantum period to try to dephase those signals relative to the ones that we wanted to measure, which was the glutamate signal. And so this is a fairly simple pulse sequence that we published a couple of years ago, which is just a conjoining of a double quantum element with a read or dephasing element. So this is a pretty standard double quantum sequence, conversion, reconversion, and then followed by a read or dephasing element on the N15 signals prior to the detection of the signal. And this worked pretty well. We demonstrated this with ubiquitin. So you can see the two data sets of this double quantum, single quantum looking experiment in the presence of the N15 pulses and in the absence of the N15 pulses without the dephasing. And what you see is that if you plot the S over S naught values for the different sites, as we retain quite nicely the signals of the aspartate and the glutamate signals, but we dephase nicely the asparagine and the glutamine signals as well as the backbone sites. Some of the glutamine signals require a little bit longer dephasing due to some dynamics that are present. All right, so now we can apply this method to look at EMRE. And again, what we did is to reconstitute EMRE into lipid vesicles. We use DMPC lipid vesicles. We've also done E. coli lipid vesicles and observed similar effects. So this data set is shown at pH 5. And what you can see is we have a really nice separation of the glutamate 14 signal relative to the other two anionic residues that are not essential in the transport process. And I mentioned this before, but EMRE is an anti-parallel structure. It's also asymmetric, which means the two monomers within the structure in principle give rise to different chemical environments in two separate sets of chemical shifts, which was first observed in Katie's lab a few years ago. And what we've shown in triple resonance experiments is that both of these sites both of those two signals are sort of underneath this peak that's shown in this red box. And this location of the carboxyl signal in this direct dimension really corresponds with a protonated glutamate signal. We could then raise the pH value to pH 10, and we observe that this protonated signal goes away, and what we see in our spectrum is predominantly a deprotonated signal. Now, of course, the middle pH is the most interesting to try to get some insight in terms of whether the two glutamate residues in the protein have different protonation states in a fairly direct measurement. And what we did then is to change the pH to pH 8. And what we find is that what we observed was one signal was appearing at the protonated position and the other signal was appearing at the deprotonated signal. So actually, Katie's group had first observed the idea of two macroscopic pKa's in EMRE. This evidence was the first direct evidence directly on the glutamate carboxyl side chain. What we wanted to try to do was to observe whether indeed one of the sites was stemming from monomer A, deprotonated, and the other site was stemming from the protonated form of the protein. So we use something called a heterodimer approach. And this heterodimer approach involves mixing a wild type EMRE sample with a single conservative site mutation. And the idea is that in wild type EMRE, we kind of intrinsically have 50% of the signals as B, 50% of the signals as A, because both of the free energies of this state and this state are the same. But if we mix it with a mutant, what we could potentially do is because the contacts in this asymmetric dimer are different, perhaps we could perturb the free energy so that we could bias the equilibrium. And really what we're doing is just changing this equilibrium constant by doing mixtures of EMRE samples with respect to mutants. So we add usually an excess of the unlabeled species combined with the isotopically labeled species that we want to look at. And this approach works pretty well. I'll show you some oriented solid state NMR data, Pysema data, just to demonstrate the idea. When we have wild type labeled with just N15 threonines and carry out these Pysema experiments, what we see are that the 5 tyrosine peaks in the spectrum give rise to exactly double the number of peaks. So you can see that the peaks are labeled as monomer Bs and monomer A, but each of those tyrosines shows 2 peaks per residue. But when we mix the wild type that was isotopically enriched with those same N15 threonines with the mutant, we can then affect this equilibrium constant such that we see 5 intense peaks in the spectrum. And those 5 intense peaks are due to the fact that we're really biasing this equilibrium toward one of the states. So we really wanted to apply this approach to trying to separate monomer A and B and assign protonation states to the 2 monomers within the E-Marie dimer sample. So this was, again, the wild type sample that I showed you before. When we then mix isotopically enriched mutant, which is this fairly conservative mutant, L51I, with E-Marie, we see only the species corresponding with the deprotonated glutamate, whereas when we mix, prepared in the opposite way, the N15C13 E-Marie sample mixed with unlabeled L51I sample, we see only the protonated species in this. So we could make the assignments based on the heterodimer effects to be monomer B and monomer A populations. So of course, this brings up the idea, if there's 2 macroscopic PKs, that there may be electrostatic coupling between the sites, right? So the idea is that if you deprotonate monomer A, that could affect the PK value for monomer B. And you can kind of draw this electrostatic coupling cycle, which is well known. So we could carry out our solution NMR experiments then by titrating a lot of different pH values in heterodimers that contained 2 pKas, the wild type with the L51I. So in that dimer, there's still 2 glutamate residues. Or we could carry out experiments where we mixed wild type with E14Q. So a site that couldn't undergo acid-base chemistry over reasonable pH values, with the hope that we could get sort of microscopic-like pK values for the different transitions. And so it turns out that the wild type E14Q gives a very strong bias in the spectrum as well. What we see is when we label isotopically enriched E14Q, that gives us very intense monomer B signals, whereas wild type gives us very intense monomer A signals. This relative conformation, the bias in the conformational equilibrium is more significant when the only glutamate residue in the dimer is deprotonated, and it's less pronounced at the low pH when, as you might expect, where the glutamine is, if maybe, let's say, a reasonable mimic of the protonated state of a glutamic acid. All right, so we did pH titrations then using N15 TROSY-based experiments, proton-detected experiments, and we used heterodimers consisting of the 2 glutamic acid 14 residues or 1 glutamate 14 residues. And what we observed was this nonlinear trend, which again had been observed by Katie's group a few years earlier, albeit with some technical challenges that happen in the wild type sample that undergoes a change in the conformational exchange rate as you go from low pH to high pH, which is a separate interesting story. But in these experiments, we could see just the single monomer A or single monomer B, and what we observed was this nonlinear trend, which was indicative of a more than 2-state model, indicative of 2 macroscopic PKs. However, when we do the same kind of experiments, same pH titrations with a E-Marie dimer consisting of only a single glutamate 14 residue, we see a fairly linear behavior, which is indicative of a single PK. So we could fit all of these curves, and of course, it wasn't just the sites that I'm showing you, but those are just example sites. We could obtain 2 macroscopic PKs for the glutamic acid 14 residues of 7.2 and 8.4. However, when we did the fitting to the 1 glutamic acid 14 residue, we only obtained a PK value of 7.2, which matched with the more acidic glutamate residue in the 2 PK titrations. So getting back to this electrostatic coupling model, we then believe that we have a PK value that corresponds fairly accurately to monomer A, which is 7.2. We also wanted to look at this other PK corresponding to monomer B. And that was a little bit more tricky of an experiment that we devised. But the idea is that we would mix wild type with a double mutant that contained both of those 2 mutants that I told you about before. So one of those is replacing the glutamate 14 with a glutamine, and the other was the L51I sample. And the idea is that the L51I would have a predominant effect at the low pH values in biasing the conformational equilibrium. But once we deprotonated the only glutamate 14 in the sample at high pH values, then that mutant would have the larger conformational preference. So if we labeled wild type, we would see B at low pH. If we then went to high pH, we would see the labeled E-emery in monomer A. And the idea of this is then that we could probe the pK of monomer B, and then this equilibrium would simply switch over. So the readout will be the relative signal intensities of monomer A and B peaks as a function of the pH titration. So a little bit complicated kind of experiment, but it turned out that it actually worked. We could go to low pH, and in low pH with wild type labeled, we only observe monomer B peaks, or predominantly observe monomer B peaks. We always have the statistical mixing issue. So low in the noise, we see 2 peaks, but the predominant species is monomer B at low pH. But when we go to high pH, we then see only monomer A, and therefore it's the transition point at which we can then do the fitting about, and that's what we did. So we plotted the population, really it's just the ratio of intensities. So it's an estimate, but we could fit this curve and we could get a pK value of 8.3. So now we have pK value, which is as good as I think we can do experimentally in terms of measuring a microscopic pK for the different monomer A and monomer B sites. And what we got was 7.2 and 8.3. Now those match surprising, well, within experimental errors of the 2 macroscopic pKs that we could measure in the 2 pKa heterodimer samples, which suggests that there's really independent pK values. So they're not coupled. So it's really proceeding through this bottom pathway where we deprotonate, we create this asymmetrically protonated state where monomer A is deprotonated and monomer B is protonated. And then at that second pK of 8.4 that was measured from that macroscopic pK measurement, then corresponds with the second deprotonation. So based on some of the measurements that we've done and several that have came out of Katie's group as well, we could propose an exchange model. And that exchange model, if we start in the upper left, is the doubly protonated state. When we deprotonate preferably at the first, at the monomer A site. Then that state we believe is fairly stable state. That is then ready to bind on the cytoplasmic side of the membrane drug. Katie's group showed that when you can indeed bind drug to a single protonated species, that does have the effect of further lowering that higher pK, that 8.4 pK value. And then you can deprotonate, you then flip, so you can kick off the drug on the other side, and then you can go through the same steps in the backward direction. So we envisioned that this single proton-bound species was really a very important state to understanding proton exchange, the lack of proton leakage across the membrane. The idea is that if this state, if this single protonated species, deprotonated in monomer A, underwent flipping, then what we would do is we would simply uniport a proton across the membrane, and it would dissipate the proton gradient across the cellular membrane. But that hadn't been observed in experiments. So I'll walk you through a little bit of why we think this asymmetric protonation state leads to a unique state that when, if it's uncoupled, the 2 pKs are uncoupled that we measured, can be an important idea. So the idea of this conformational flip is then if you have both protonated, that the rate constants of sort of going from this inward open to the outward open are by definition the same. The states are exactly the same, at least under a not asymmetric membrane environment. So those are identical. But the singly proton-bound state is not the same if the proton doesn't hop from one site to the other site, one glutamate to the other glutamate. And the reason is because this deprotonated species on B is then relatively unfavorable relative to this deprotonated species on monomer A, which we showed in the E-emery E14Q E-emery titrations. So to kind of emphasize this point, if this doesn't flip, we lead to less proton leakage. Essentially, this is a kinetically stable state, which can then bind to drugs and doesn't just undergo the flipping. But if we assume that the proton were to jump from one site to the other site in so-called hopping mechanism, or even something that involves just not fully transferring the proton over such that we get to this state, we then end up with a state on the right case that you would end up with a lot of proton leak, which doesn't seem to happen in the biological system in proteoliposome assays. So we think that this hop-free mechanism provides an explanation through independent PK values of why the proton leakage doesn't happen in the cycle. Also with structural information that's come about and others that we're working on, the two glutamate residues we believe are quite distant, somewhere between 10 and 15 angstroms apart from one another, which further maybe underscores the idea that they're not strongly coupled in congruence with our NMR data. And very, very lastly and briefly, I know I'm over, I want to tell you just about how we can target glutamate and aspartate residues. And what we worked on is a different efflux pump from MRSA, gives resistance to fluoroquinolones. We solved the structure in collaboration with Danang Wang's group. Doug Browley did, a PhD student in the group. And we had to solve these in complex with FABs. And we solved two structures to get the mass large enough to solve the structures with cryo-EM. And what we observed was that both of the CDRH3 loops on the antibody that were diversified in the screen inserted into the pocket. And if we zoom in, these loop residues shown in blue make very beautiful contacts with two anionic residues, glutamate 222 and aspartate D307. So this tryptophan arginine motif is really critical. And what we did is to essentially make peptides mimicking the loop that inserts in, something like this. And we showed that this could inhibit MRSA growth when used in combination for fluoroquinolone. So not only are those glutamate aspartate residues playing critical roles in the overall transport cycle, but we think they're targetable sites with peptide-based peptidomimetic inhibitors. So with that, I'll thank you for your attention and take any questions.
Thanks, Nate. That's absolutely beautiful work. So we have time for a quick question. I'll ask one. What I didn't get from the mechanism was how this efflux pump would maintain concentration gradient, right? It looked perfectly symmetric. So why aren't things coming in as fast as they're going out?
Well, actually, Katie's group has been pursuing a very interesting hypothesis, and she has some nice data to support that. But you actually can get things that would be imported in. And this is thought to be some role of, so a different, rephrasing your question, a different way to ask the question is, what is the real function of these efflux pumps? Because it probably wasn't antibiotic resistant. So they're probably involved in metabolite movement across membranes. Actually, Randy Stockbridge's group has shown that a different subgroup of transporters in Emery's family gives rise to guanidinium transport. So quaternary ammonium compound. So you can have import in, you can have import out. It depends on the pH environment that you have across the membrane and the available substrates on the different side of the membrane. And some mutants play a role in that as to
Okay, so the proton gradient plays an important role in that. The
proton
proton
gradient
gradient
plays a
will play
role.
an important role. Yeah, I see. Yeah. Katie?
Hi, Nate. That's beautiful work to assign the actual PKAs to glutamate 14A and B. I love it. At some point, I have some questions with the hopping mechanism about the relative rates of alternating access versus on-off of the proton, even from like bulk solution, and how that would be restricted to sort of make this model really prevent proton, just protons are so tiny.
Yeah, that's a good question. So I think Katie may be saying, even if this happens, the specific rates of protons being pumped off might actually be so slow to not overall influence the transport cycle. Is that basically your question? A little bit?
Yeah. It's the relative rates of protonation versus alternating access. Yeah.
Oh, yeah. So the relative rate of alternate. So I think what you're saying is the alternating rate of access is quite slow relative to the overall proton leakage. So that can certainly be another way to prevent the overall leakage of the proton across the membrane. If you think about this statistically, that the proton coming off of this side might be quite slow relative to this. Are you saying that the proton should come off really, really quickly, or are you saying it should come off slower? Yeah, so that's actually kind of interesting. We've seen some we've observed some things by solid state MR that suggest the proton doesn't come off quite as quickly as you would expect into bulk kind of media. So I'm not quite sure about that proton off rate. I know we've discussed this at one point.
We should talk more because we have some funky data too.
Yeah, that's an interesting point. Thanks for your comment.
And we have another question.
One more. Okay, hi. Have you considered raising camelid nanobodies, heavy chain nanobodies to the MRSA efflux pump?
So, yeah, so what we did do is we did phage display in collaboration with Shohei Koide's group. So it's a different, maybe that you have some different ideas, but the alternative way of generating synthetic binding proteins.
Yeah.
That's what we
No,
did
you
do.
probably, you use phage display here, but the camelid, the heavy chain nanobodies have more utility in number one, they're extremely resistant to degradation, looking forward to a possible clinical application. And number two, enhanced tissue penetration. So that might be something to look into down the road.
Yeah, it's a good suggestion. I should say, one thing I zipped by really quickly here is our goal here is to take a synthetic binding protein on the left, whatever that happens to be. Maybe it's a camelid antibody, a monobody, whatever that happens to be. In this case, we're using FABs, but to miniaturize the part that inserts into the pocket so that we can have the absolute smallest portion of the regions from those synthetic binding proteins that can insert. In this case, we use simple peptides. So we only used a 12 residue peptide corresponding to the loop that was then used in these dose-dependent experiments.
I
with
see,
MRSA.
I see very well where you're going with that. I know that, but the question is, how would such a peptide, if you were going to use it in a clinical application, is it going to make it to the target intact, or is it going to get chewed
Yeah,
up?
so that will always be a potential issue that we'll have to get through with, maybe biofilm formation of how, ideally what we want to do is, right, this was the first step. We said the key contacts were made by the loop insertion. So we made the 12 residue. Ideally, we want to make that as small as possible and only retain the key parts to make it as small molecule like as possible for the purposes that you're saying. Better, penetration across whatever biofilms or whatnot.
Well, for in vitro purposes, sure, but just looking down the road, the camelid antibodies might be something, they might have some utility. Thank you.
Sure.
Okay, thanks, Nate, and thanks all the speakers for a wonderful session this morning. Thanks, everyone. That was great. That was a wonderful session.